Hello everyone, I'm Dr. Deepika Malik. Welcome back to my channel. In this video, we'll discuss ELISA, that is Enzyme-Linked Immunosorbent Assay. It is widely used laboratory technique for detecting and quantifying specific proteins, antibodies, antigens, hormones, and other molecules in a biological sample. The ELISA principle is based on the specific binding between an antigen or antibody and its corresponding antibody or antigen while utilizing enzymes for signal amplification. Substrate addition. A substrate for the enzyme is introduced into the well. If the enzyme is bound to the well due to the presence of the target molecule, it will catalyze a chemical reaction with the substrate, producing the detectable signal, usually a color change or luminescence. Signal measurement. The intensity of the signal, which is directly proportional to the amount of bound enzyme-linked antibody, is measured using a spectrophotometer or a specialized ELISA plate reader. Data analysis. The absorbance or fluorescence of each well is compared to a standard curve of known concentrations to determine the concentration of the target molecule in a sample. As you can see here in the image, 96 well plates or 8 well strip tubes are used. Both these plates and tubes are made of polystyrene. The 96 well plates provide plenty of spaces for the determination of optimal dilution for the reaction. In the meanwhile, 8 well strip tubes are better for the finely tuned assays. Enzymes for ELISA. Enzymes used in ELISA play a crucial role in detecting the presence of specific antigens or antibodies. They are conjugated to detection antibodies and generate a detectable signal when the target molecule is present, as you can see here in the diagram. Here we have a capture antibody to which a target antigen binds, and now we are taking another antibody to which an enzyme is linked. And now when we provide the substrate reacts with enzymes producing a colored product, giving an indication that it has been a positive binding. Now some of the commonly used enzymes in ELISA include, as you can see in the table, horseradish peroxidase, which catalyzes the conversion of chromogenic substances into colored products. Substrate used for this is TMB or ABTS. Alkaline phosphatase, which hydrolyzes phosphate groups from substrates, resulting in a yellow color change. The substrate used for this particular enzyme is B-nitrophenyl phosphate. Glucose oxidase, it catalyzes the oxidation of glucose in the presence of oxygen, producing hydrogen peroxide. Hydrogen peroxide can then react with a chromogenic substrate to produce a colored product, and the substrate used for this is OPD, which is O-phenylene diamine dihydrochloride. Then beta-galactosidase is used less frequently but can be used in colorimetric ELISA. It hydrolyzes colorless substrate into yellow products. The substrate used for this is orthonitrophenyl beta-galactoside. Acetylcholinesterase, which is used for acetylcholine detection. It catalyzes the hydrolysis of acetylcholine to produce theocholine, which reacts with Elman's reagent DTNB to produce a yellow color change. The substrate for this enzyme is acetylcholine. Then we have luciferase, which is used in chemiluminescent ELISAs. It catalyzes the oxidation of a substrate, resulting in the emission of a light. The emitted light is measured by a luminometer. The substrate for luciferase is luciferin. The ELISA giving signal in form of light is known as chemiluminescent ELISA. So the enzymes are exposed to reagent which cause them to produce light or chemiluminescence. It is a process where light is produced as a result of a chemical reaction. It occurs when chemical reagents undergo an enzyme catalyzed reaction with oxygen. A chemiluminescent substrate such as luminol is added to the microplate well. If the enzyme labeled detection antibodies bind to the target molecule, it will catalyze a chemical reaction with the substrate, producing light emission known as chemiluminescence. The emitted light is then quantified using a luminometer or a specialized plate reader. So as you can see here in the diagram, we have a capture body to which the analyte of interest binds, and to this enzyme labeled antibody binds, and when there is a reaction, there's a production of light as a signal giving a positive indication. Types of ELISA. ELISA tests can be classified into four types depending upon the different methods used for binding between antigen and antibodies, namely direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Now let us discuss all these types one by one in detail. Direct ELISA. Here antigen is coated to the microtiter well. The primary antibody is directly conjugated to the detection enzyme. This means direct ELISA uses a single antibody. The enzymatic reaction on the substrate causes a color change in the medium, which depends on the type of substrate and the enzyme used for the reaction. This facilitates the detection of samples with the target antigen. The steps of direct ELISA are listed below. Coating the surface of the plate with the sample, as you can see here in the diagram. Incubation of the plate with the enzyme conjugated primary antibody. Washing for the removal of unbound antibodies from the plate. Then addition of a substrate required for enzymatic reaction. And then finally detection of the signals produced from the plate. Direct ELISA is more suitable for the detection of the high molecular weight antigen amount in a sample. The fewer number of steps in the ELISA methods makes it a faster assay. However, due to the cost and time incorporated with the labeling of the primary antibody with enzymes, the use of direct ELISA is quite rare. On the other hand, it produces adverse effects on the target antigens as well. Indirect ELISA. It is another method of ELISA which uses two antibodies for the detection, primary and secondary antibody. Due to the availability of enzyme-conjugated antibodies, the indirect ELISA method is more often used in immunological assays in the detection of parasites, bacteria, and virus. On the other hand, several secondary antibodies can be bound to the primary antibody in the plate. Therefore, the sensitivity of the assay is very high. The steps of indirect ELISA are as follows: coating the surface of the plate with the sample, incubation of the plate with the primary antibody, then washing to remove the unbound antibodies from the plate, incubation of the plate with the secondary antibody conjugated with the detection enzyme, then washing to remove the unbound antibodies from the plate. Addition of the substrate is then required by the enzymatic reaction and then detection of the signal produced from the plate. There are limitations to this method. There's a more complex workflow, that is, an additional incubation step is required for the secondary antibody, potential for cross-reactivity, non-specific signals due to cross-reaction with the secondary antibody can also occur. Sandwich ELISA. It helps to detect the presence of antigen in a sample. The biotin streptavidin binding is exceptionally strong, which helps in minimizing background noise and improving assay sensitivity. Sandwich ELISA helps to detect the presence of antigen in a sample. The biotin streptavidin binding is exceptionally strong, which helps in minimizing background noise and improving assay sensitivity. Now let us see the steps of sandwich ELISA. The microtiter well is coated by the antibody, as you can see here in the diagram. To prevent non-specific binding, the plate is blocked with a blocking agent, often BSA, which is bovine serum albumin or milk powder. The sample containing the antigen is then added to the well and washed to remove free antigens. Then a biotinylated detection secondary antibody, which binds to another epitope of the antigen, is added. The well is washed to remove any free secondary antibodies. Streptavidin is then added to the plate. Streptavidin has a very high affinity for biotin. In this step, streptavidin conjugated to horseradish peroxidase, which is basically our enzyme, is commonly used. The plate is then washed again to remove unbound detection antibody and streptavidin HRP. A substrate solution containing a chromogenic substrate for HRP is added. HRP catalyzes a reaction with the substrate, resulting in the development of a colored product, which can then be measured, as you can see here in the diagram. Now let us see why biotin is used as a labeling molecule. First is reduced steric hindrance. Biotin is a relatively small molecule, which means it can be easily conjugated to antibodies or other molecules without significantly affecting their binding properties. This is important because you do not want to label to interfere with antibodies' ability to recognize its target. Amplification. The biotin-streptavidin interactions provide a method for signal amplification. In a sandwich ELISA, when the biotin-related detection antibody binds to the target protein, it brings multiple biotin molecules into proximity. Streptavidin horseradish peroxidase or streptavidin alkaline phosphatase, which are added in the subsequent steps, can each bind to multiple biotin molecules. This results in signal amplification, making the assay more sensitive. Low background. Biotin streptavidin interactions are highly specific, so they result in very low background noise. This specificity contributes to assay sensitivity and accuracy. Commercial availability. Biotin-related antibodies and proteins are readily available commercially, simplifying the labeling process for researchers. Stability. Biotin-related molecules are stable and can be stored for extended periods without loss of activity. Competitive ELISA. It helps to detect antigen concentrations in a sample. The steps of competitive ELISA are: first, the microtiter wells are coated with the antigen. Antibodies are incubated in a solution having the antigen. The solution of the antigen-antibody complex is added to microtiter wells. The well is then washed to remove any unbound antibodies. More the concentration of antigen in the sample, lesser the free antibodies available to interact with the antigen which is coated in the well. The enzyme-linked secondary antibody is then added to detect the number of primary antibodies present in the well. The concentration is then determined by spectrophotometry. As you can see here in the diagram, first we have incubated the antibody with antigen to be measured. And if this antigen is specific to this antibody, it will occupy all the binding sites available on the antibody. Now, when we add this mixture in a well containing already labeled with the antigen, there will be less free antibodies which are available to interact with this red antigens. Since only few antibodies are able to bind to these antigens, therefore the colored substance produced will also be less in amount, and thus with the increasing antigen concentration, there will be a decrease in the signal, there will be a decrease in the colored product formation, giving a positive result. Now let us discuss ELISA applications. In clinical diagnostics, it is commonly used in initial screening for HIV antibodies in blood samples. It can also detect hepatitis B and C viruses in patient samples. It can identify autoantibodies associated with autoimmune disorders like lupus and rheumatoid arthritis. ELISA assays are again used to detect specific cancer-related biomarkers in serum or tissue samples. It also measures hormonal levels in endocrinology, such as detecting thyroid hormones or insulin. ELISA is also used for infectious disease detections, like for pathogen detection. It detects antigens or antibodies related to various infectious agents like bacteria, viruses, and parasites. Serology testing. ELISA helps determine past exposure or immunity to diseases like measles, mumps, and rubella. Pharmaceutical research. For drug development, ELISA is utilized to screen and quantify drug candidates, study drug-protein interaction, and assess drug efficacy. Biomarker discovery. Researchers use ELISA to discover and validate biomarkers associated with diseases or drug responses. Immunology and allergy testing. ELISA identifies allergen-specific IgE antibodies to diagnose allergies. Immunoglobulin quantification. It quantifies immunoglobulins like IgG, IgM, IgA for immunological research. Food safety and quality control. Food allergen testing. ELISA identifies allergenic proteins in food products. Pathogenic detection. It can help screen for foodborne pathogens like Salmonella and E. coli. Quality control. It monitors the quality and authenticity of food and beverage products. Veterinary medicine. Animal disease diagnosis. ELISA is used for diagnosing diseases in livestock and pets, including brucellosis and heartworm. Research and biomarker discovery. For protein quantification, ELISA quantifies specific proteins in research studies, including cytokines, growth factors, and antibodies. Cell signaling studies. ELISA measures phosphorylated proteins in signal transduction pathways. Environmental and toxin monitoring. ELISA is applied in environmental research to detect pollutants, pesticides, and toxins. ELISA assists in blood typing and cross-matching for transfusion compatibility. ELISA detects plant pathogens for crop protection. Drug and alcohol testing. ELISA screens for drugs of abuse in urine or blood samples. Alcohol biomarkers. ELISA measures alcohol metabolites in forensic or clinical settings. The last slide shows a comparison between radioimmunoassay, immunofluorescent assay, and ELISA. The principle for radioimmunoassay, it is based on the competition between a radioactive labeled antigen, that is tracer, and an unlabeled antigen from the sample for binding to a limited number of antibodies. The amount of radioactivity in the bound fraction is inversely proportional to the concentration of the specific antigen in the sample. Immunofluorescence assay uses fluorescent labeled antibodies to detect the presence and location of specific antigens such as antibodies or pathogens within cells or tissues. It relies on the binding of fluorescent antibodies to target antigens. ELISA employs enzymes as labels. It can be performed in various formats, including direct, indirect, sandwich, and competitive ELISA. In sandwich ELISA, for example, the antigen of interest is captured between two antibodies, one immobilized on a solid surface and the other enzyme labeled. Labeling. In radioimmunoassay, radioactive isotopes, example I-25 iodine, are used as labels, making it radioactive technique. For immunofluorescent assay, fluorescent dyes or markers are used for labeling antibodies, making it a fluorescent technique. In ELISA, enzymes, example horseradish peroxidase or alkaline phosphatase, are used as labels, and the assay produces a colorimetric or luminescent signal. Detection. Radioactivity is measured using a gamma counter in case of radioimmunoassay. Fluorescence is detected using a fluorescence microscope or a fluorimeter. Colorimetric signals are measured using microplate reader in case of ELISA. Safety. Radioimmunoassay involves the use of radioactive materials, making it potentially hazardous. Immunofluorescent assays are generally safer since it doesn't involve radioactivity. ELISA are safer than radioimmunoassay but may still involve handling chemical reagents and biohazardous materials. Sensitivity and specificity. Radioimmunoassay can be highly sensitive and specific but may be limited by cross-reactivity with structurally similar molecules. Immunofluorescence assays offers good sensitivity and specificity when used with appropriate antibodies. Sensitivity and specificity can be tailored to the assay design of ELISA and the quality of antibodies used. Thank you for watching.